Hello wonderful viewer. At the end of this video, you would be able to understand the transcription cycle of a prokaryotic RNA polymerase. So watch to the end. The first one here, this one is the sigma factor, and this one is called the core enzyme. This enzyme without this one is called core, but when this sigma factor attaches itself to the core enzyme, everything becomes a holo enzyme. So first of all, the holo enzyme will recognize a promoter on the DNA, a promoter sequence on the DNA, and the sigma factor is the recognition, is the one that will provide the recognition for the holo enzyme to attach itself to the DNA. So when it is properly attached to the DNA and oriented properly, then an unwinding starts to occur. Very small part of the DNA begins to unwind. Okay, so when it unwinds, then the RNA begins to synthesize. So the sigma factor, which is attached to the promoter, would help the DNA molecule to unwind. After it unwinds, you will see that a piece of RNA would begin to synthesize. Immediately this piece of RNA begins to synthesize, we would have our sigma factor detached. It will drop off, leaving the core enzyme, our core enzyme. So still with the DNA strand, and the RNA continues to elongate now, but here it continues to elongate as the core enzyme moves along the DNA molecule, and the chain keeps on growing and growing and growing till it forms a hairpin. When it forms a hairpin, it will signalize termination. It will mean that it has gotten to a point whereby we have to stop synthesizing for the DNA to be detached from the core enzyme and for the RNA to also be released. So this is the hairpin. This hairpin signalizes termination. So here, what we have in totality, we would have our enzyme, the sigma factor. Without the enzyme, the RNA polymerase is called a core enzyme, but when both of them are together, the sigma factor and the polymerase, the core polymerase or core enzyme is together, it is called holo enzyme. So the holo enzyme will find the promoter sequence on the RNA, bind to it and unwind it a little bit. When it unwinds, then the nucleotide sequence begins to synthesize. If it synthesizes the first 1, 2, 3 up to 9 bases and there is no error, then the sigma factor detaches itself from the process. Then the chain begins to grow as the enzyme moves along the DNA, molecule downstream. So the chain continues to grow until it forms a hairpin, which signalizes termination. Then our RNA will be released and our core enzyme too will be released from our DNA. So that is how transcription is done in prokaryotic cell. Thank you for watching.